So my talk is a little bit more broad. The title is Accomplishments and Prospects in High-Resolution Imaging. And of course, I'm going to talk about columns and layers in particular. And here's the outline of my talk. I want to touch base a little bit on the early history of pursuing columns and layers, all challenges and problems, some solved and some are still with us, as Seiji has outlined very clearly. And I want to look at the future, not now, so to speak, start looking at the future maybe sometime when we were in the middle of this decade, like 2014 or something like that. And then I want to summarize some of the recent results that are coming from CMRR that will be relevant for you. Now, many of you may say, okay, Kamil, why do you have to go and review the history again, especially since you're probably going to show some of the same slides you always show when you talk about high-resolution fMRI. And apologize for that, but you know that fMRI has been with us for about 3 decades now, and some of us are getting very old. We have a hard time remembering things, and many of you probably in this audience listening to this talk are very young and may not even have been born at the time of fMRI was invented. And so it is good to essentially review a little bit where we were and while we were talking at the same time where we are going. And I want to also mention that this is not a review lecture of the field, so I'm going to be really focusing on the effort from CMRR. And finally, I think, unfortunately, as people know, I tend to give long lectures, and this will be somewhat of a long lecture. And it may bore you, in fact, some of the stuff that I will cover initially because of the fact that you have seen it before, most likely. But there are some interesting things I'm going to show towards the end of the lecture, and you may be interested to wait for that, or you can take a snooze if you like at the beginning, and then, but please, you may want to be awake when I go towards the end. I mean, maybe just to give you a hint, I mean, you may want to see, for example, how you can get a factor of 2 or 3 improvement in the contrast to noise or signal to noise of your fMRI results. Or you may want to see first human results obtained at 0.5 millimeter isotropic resolution. So just a hint that there are interesting things coming down the pipeline. So when fMRI came into existence, of course, Uğurbil and Vizioli had already demonstrated the orientation tuning of cells in the V1, but human fMRI, when it came to existence, of course, took in the direction of human applications and human neuroscience, and at that time, human neuroscience was really not thinking about cortical columns or layers. I mean, they didn't have any tools to study that in the human, and this kind of consideration was very far away from their vision. And but Uğurbil and Vizioli, when they demonstrated this with electrophysiology, had some idea about, well, there's some organization, but really the first hint or the first demonstration of the orientation, how the orientation tuned cells are organized in the visual cortex, came from this paper from Amiram Grinvald's group using intrinsic signal, optical imaging of intrinsic signals, showing the pinwheels and the distribution of the orientation domains along them. And they said here in this paper, said we find that cortical regions that respond best to one orientation form highly ordered patches rather than elongated bands. So this is really the demonstration that these organized patches, how they exist. So these are intrinsic signals. They are hemodynamic signals. So in some ways, they are very comparable to fMRI, not exactly as you know, but comparable. So of course, it raised the question, especially in our group, as to whether fMRI can ever get to do something like this and image organizations like that. But there were also other papers from Amiram which raised questions about fMRI's ability to be able to do something like that. And not just Amiram, but there was also a big skepticism about fMRI's ability to get to that kind of a resolution. This is a paper by Malonek and Grinvald in 1996, where in the abstract, I would read this here, Sensory stimulus of cortical columns initiates tissue hypoxia and vascular responses that occur within the first seconds and are highly localized to individual cortical columns. This is the so-called the dip argument that became quite prominent for a while. And then, but they say, however, the later phase of the vascular response is less localized, spreading over distances of 3 to 5 millimeters. And that is ominous as a prediction for fMRI, in fact, because we are usually looking at the later response. And at the end of this paper, their concluding sentence is, this large mismatch between oxygen consumption and supply probably underscores the importance of adequate oxygen supply, watering the entire garden for the sake of one thirsty flower. And they also reference this abstract by Bob Turnell and Grinvald himself. And so our first, one of the very first studies, this is Sanjay's group in CMRR doing this work, was to actually ask the question whether we can, in fact, look at imaging these vice orientation columns with CBF. This is not bold, this is a CBF experiment. And because the major problem, the issue at that time is really, is the biological processes involved, the hemodynamic processes involved, are they really accurate at the level of columns and, of course, later layers? But this is a column question at the present time. So he did these orientation column mapping in the cat cortex, just like Amiram was doing. And they found, this is first author is Tim Gong. They found, of course, CBF responses, and there were areas that responded and showed very high CBF responses with a particular orientation presentation. And other areas also showed some CBF responses, but considerably less. And if you go to the orthogonal, and then, well, you can then make a map from this because there's a difference between these two responses. And the map, in fact, then presents something like the orientation domains. It's a single condition map. I want to emphasize that. And then you, of course, go to the orthogonal presentations. And in the orthogonal presentations, the areas that show the high response initially and now start showing the low response, and vice versa. And of course, then you get these complementary maps and the map of orientation selective domains. And so this particular work, in my opinion, really, this was a very critical work at the time, because really there was quite a bit of controversy about the specificity of the hemodynamic response. And this is the first work that actually said, okay, CBF is regulated at the level of cortical columns. And the CBF response, there was a point spread function that was measured 490 micrometers. And I keep, by the way, writing perfusion because the CBF that is measured by fMRI in these studies is really a perfusion measurement. It suppresses large vascular contribution. It's a detail that I won't be able to go into more, but of course discussed in the papers. So does the brain water the entire garden for the sake of a thirsty flower? And the answer is really not really. And what I suggest is that the brain waters the thirsty flower while it generously sprinkles around it as well. And of course, this is CBF, but we knew at the time that there's a problem with BOLD, because if you do now gradient recalled echo BOLD in the same study, in the same model, you get images like this, where it is really the large vessels that dominate on the surface this particular mechanism. And you see, for example, instead of seeing these nice columns, you see these sagittal sinuses, the major and the largest contributor, and irrespective of the orientation of the gratings that you are presenting. I said we knew about this because we had actually very early on noticed this. It was, in fact, when we noticed this, and in those days, the experiments were done with not only with everybody in this, Seiji would come, and Ravi was my postdoc at the time, and we would actually sit and do these experiments, look at the images as they were coming out. We had noticed these the vein effects, and it was actually somewhat of a disappointing observation for us, because when we saw them, that we knew that there was going to be some trouble in terms of getting to the information that we really wanted to get at. But this work conducted by Ravi finally said, utilizing high-resolution images, two distinct regions ascribed to microvasculature and visible venous vessels were identified as giving rise to the signal increase. So this is a demonstration of the presence of the large vessel contribution and large vessel coming from most of them in the peel surface. So, yeah, that we are talking about, there is this kind of a contribution at 4 Tesla, but there's also biggest contributions coming from there. But many of you probably don't remember this work very well. It is very old at this point, and you can't even get a decent PDF copy if you download it because they were not available at that time. You can download this, but you can see that it was some sort of a scanned copy that is now available from the journal. But most of you probably know this vessel effect from this work, from MGH, from Polomini et al. And what they showed was, okay, if you were to print an M on the cortical surface, M for MGH, but maybe for Minnesota, if you print, take advantage of the retinotopic representation in the cortex, you can print an M on the cortex. And this is a function of layers. And you can see that in the outer, near the peel surface, the M is not very well defined. And it gets better and better defined as you go to the, near the white matter. So this is the same vessel problem. In some sense, it's an old story, but it's actually presented in a very new, attractive way after all the developments that have occurred when we can actually visualize these things in the surface and we can get high-resolution images in the human brain, etc. But I want to just point out that this is actually a differential image, what they are showing. See, you can see it here. So differential image is better than single condition images. So many of the problems are kind of canceled, but they don't cancel each other very much. These are the single condition images. So their differential image was they were either showing this pattern, which translates into the M, transforms into the M on the cortex, or they were activating this, or they were activating everything else but that. Those are the differential conditions. So we really should look at the single condition like this. So this is, in fact, now what you would see if you were to present this versus nothing. And it's really pretty bad in the cortex. I mean, the surface really bad. And you start getting a hint of the M in the inner layers when you go into in depth. So, but okay, even though we had anticipated a problem like this because of Ravi's early work, we thought that, okay, there should still be voxels where you can actually maybe look at columnar responses. So very first effort on columns, again, from Ravi, and when he was in Minneapolis and subsequently went into Roberts, he then to Canada, Roberts Research Institute, where he did many more column experiments. But this one from Minneapolis, 4 Tesla, it was published in 1997. I have to admit that the images did not look so good, columnar, but we did have time courses that suggest that we are actually getting responses to ocular dominance columns. And first column images came from Kang Cheng, from Riken in Keiji Tanaka's lab, and they had actually a clever idea because the resolutions were not good enough in those days, and they actually had a subject. They found the subject, as you can see, with a very flat calculum fissure, so you didn't have to worry about all the convolutions of the cortex in the human brain. You can just take a single slice and hope to see what the columns are like. And they did present these images. These are differential maps, again, important differential maps, with varying p-value threshold. So they, look much better than in our images. You can see actually the presence of ocular dominance columns, but still, it's not really super. So what did we do? In our case, we went to 7 Tesla to be able to reach this columnar scale robustly. And this is a picture of this magnet being installed in 1999 in CMRR. And the reason we went to this is really based on this work that we published in 1993, which is a modeling paper of all the bold responses. And in this modeling paper, in fact, there's a prediction that you have, of course, vessel contributions coming from different diameter vessels, but the contribution of these capillary or microvessels, should increase quadratically, whereas the Bold effect from these should increase linearly. So you can actually say, well, let's go to high fields, even though in those days there was a lot of questions about high fields, like does the signal noise increase, etc. But we were courageous enough to try to test this. I want to just emphasize, this is what maybe the contribution of going to 7 Tesla in CMRI should not really be minimized. This was not an effort from major manufacturers like Siemens and GE or Philips. They didn't really want to touch the stuff in those days. They were later on dragged into it. But we were convinced about or interested enough in it enough to be able to work with David Rayner, who built us this 7 Tesla magnet. And the rest of the system was actually put together by us, mainly buying components from different manufacturers or engaging different manufacturers like to build us amplifiers that will work at 300 megahertz, and a lot of components also built in-house, including digital receivers for multi-channel receive and RF coils. And there aren't, if you think about it, there aren't that many labs actually. Can you think, okay, let's do say a 11.7 Tesla and I'll build the console and everything like that. So this was a major effort from us. And of course, we now have this meeting because of the fact that 7 Tesla really ushered in the ability to do human imaging at the columnar level. And we finally did achieve, this was really something that I really worked for about 2 decades, so I have a hard time not covering the history, as you can imagine. It is very dear to my heart. It's a major part of my career was committed to this. So we did get at 7 Tesla really robust ocular dominance columns, and these are single slices using the same trick that Riken investigators used. This is a subject with a flat cortex here. This is work by Isa and Amir Shmuel. And in this case, Isa was responsible for acquiring the data, and Amir and his people really did all the analysis for this work. So really nice reproducible columns. I'll show you a little bit more of that. And then the orientation columns for the first time in the human primary visual cortex, and showing the relationship between ocular dominance and orientation columns. So now, looking at the optical imaging data, intrinsic signals from the monkey and human data, there are many characteristics that you can actually see. For example, the linear zones perpendicular to the ocular dominance columns here in the monkey, in the human, the pinwheel centers. This is Amiram's work saying, oh, they tend to appear in the middle of the ocular dominance columns. Yeah, they are in the human, etc. But sometimes we still get people somehow questioning the veracity of these results. I have to say that they don't read the paper very carefully, because there's a very, very nice, convincing experiment that we presented in this paper. And that is that when you are looking at orientation domains, you have these pinwheel centers, and some of them go clockwise, and like this one here, and the other ones go counterclockwise. So if you are presenting the stimuli, let's say in this clockwise, you will get clockwise, the red pinwheel centers, clockwise rotations, and then the others you will get counterclockwise rotations. Well, there's a very nice experiment. You can now change the presentation, then you go the other direction, then these guys should switch. And in fact, this is what happens. You can see that the reds become green, the green guy becomes red. If you can follow these rotations, that they have now switched direction from clockwise to counterclockwise and vice versa. So we are very happy with these results. And of course, for many years, and a lot of MR people, fMRI people talking about where fMRI was going would show these results. And then, of course, these days, there are many other columns, layers in the human that we are seeing. But I wanted to come back and look at this. Well, as I mentioned, these are reproducible results. This is ocular dominance columns again. This is not a map of ocular dominance column, rather it's a reproducibility map. It of course shows the presence of the ocular dominance columns. It's an overlap of 2 days. We actually did not only spin echo, but we did gradient echo as well. And you can see that in the gradient echo, as expected, you see some of these columnar structures, but in other areas you don't, because predominantly of the large vessel effects. So, coming to layers, CMRR has a large history in layers. This is data that Seiji showed. Seiji was the first person to start asking questions about layers when he was in CMRR. He showed this slide. He has a better slide than this is from the paper. I have to get that slide from him. But yeah, we have quite a long history in layers as well. And this is another work by Noam Harel, where he looked in the cat cortex now with histology and looking at vessels, as well as the histology showing the fact that, as Sanjay described, the gradient echo tends to result in contribution primarily on the surface, and spin echo is in the deeper layers mainly, but somewhat more diffuse. And there has been, and then the first human paper actually looking columns and layers. This is work together with our Maastricht colleagues, and look in the human MT area, organization of axis of motion selected features in human area MT, and and Rainer did a beautiful job of actually analyzing and segmenting this data. Well, actually, I should not just give credit to Rainer. Jan Zimmermann, who is now, in fact, faculty member at CMRR, and who is just published. I think it may have appeared in press already, but if not, very soon, in Neuroimaging, the first monkey studies at 10.5 Tesla. But nevertheless, this is first high resolution looking at. These are Grace sequence from working with David Feinberg, and shows both columnar and layer structures. There are a lot of other layer work. You've, I'm sure, seen this maybe this is the first paper showing that there are cognitive effects that you can detect across layers in the human. This is work by Cheryl Allman. And this is a collaboration with Lars Mückli, really beautiful study in the human visual cortex, where you are decoding essentially from the visual cortex in this particular, from neurons that respond to this particular area. Because of the retinotopic representation, of course, you can actually just look at this area in the cortex and try to decode whether they are seeing this image or not. And when in this data, what we were able to show that feed forward, when they are looking at this, is sort of decoding is kind of flat across the cortex, but in this case, you actually still can decode, but in this case, the pattern is very different, suggesting that the feedback information is coming to the outer layers. But many, this is work, of course, really beautiful set of studies, in collaboration with Maastricht again, in the auditory cortex, going from tonotopic representation to inferior colliculus tonotopic representation, to medial geniculate body organization, and finally coming back to the primary auditory cortex, looking with laminar resolution tonotopic representation across the lamina, and even showing that actually attention modulates responses across the layer in this really nice work. So a lot of work actually since then on layers in human brain, but we are still looking at, in a lot of ways, a lot of times, unlike the experiments that Sanjay presented, where you can modulate a lot of things by invasive measures, we are relying on cognitive responses to be able to say, oh yes, we can actually maybe seeing layer differences in the human brain. So at about this time then, we started looking into the future. And that future, actually, I want to, for me, translated into trying to formulate strategies for this BRAIN initiative. This is BRAIN 2025 scientific vision document. I was lucky to be one of the 14 members who formulated this document. And in this document, there were 6 priority areas, essentially looking at the future. This was published in 2014. And it took us a couple of years and a lot of brainstorming with a lot of colleagues to come up with this document. But at the end, we came up with 6 priority areas. And one of them, number 2, actually maps of multiple scales and then called for maps for synapses to whole brain. And there was in fact, a very specific recommendation. The BRAIN initiative target for whole brain studies called for 0.1 microliter or smaller voxel volume to be achieved in MRI studies. Now, you can say, well, who put that limit of 0.1 microliter and why 0.1 microliter? And of course, it was a number that came from me, and my rationale was, okay, at 7 Tesla, we are doing very robust imaging at about 1 millimeter isotropic voxels, which is 1 microliter. And, we can aim for a factor of 10. And I thought factor of 10 is probably achievable in a reasonable amount of 10. I didn't want to put down something like 0.01 microliter. That would have been maybe too ambitious. I wanted to put something that where people can think, okay, we can try and we can likely reach it. And this is, by the way, 0.1 microliter is about a half a millimeter isotropic voxel size. So, of course, there have been a lot of efforts in many places trying to reach that with MR and other techniques as well. And I want to just give you some, oh, by the way, and of course, also in this document, there was a call for trying to understand what is the connection between neuronal activity and fMRI signals. So I want to give you a little bit of then, I keep going ahead of me in this lecture, but so there was, of course, for understanding that, but essentially we really had to rely, in this, we envision that it's not possible really to cover this whole scale. That we can cover this scale from a whole brain to cortical columns. This is orientation maps I showed you. And it's very hard to go beyond that. We are trying to go, that we can put it as an aim, an ambition to go beyond that. But we can, of course, cover the same scale with multiphoton, this lower scale, the multiphoton imaging, going to single cell responses, and as well as obtaining columnar images. These are, again, the same kind of orientation domains. This is cat, this is ferret that you can actually get by the 2 techniques. So there's an overlap between them. Sometimes I hear my MGH colleagues talking about, well, we're going to go from here to here and from here to here. One day they're going to touch. And I would like to correct them, saying that they have already touched. There's an overlap area. They touch, and we can use these 2 techniques. And I want to tell you a little bit about what is going on in CMRR using both of those techniques and trying to achieve the BRAIN initiative goals. So looking at the future, then in CMRR, we have been conducting a lot of animal studies, also human studies. The animal work I want to show you is done by this group of people: Xin Ho, Xiaohong Su, Wei Chen, Wei Su, Jodat Idiatulin, and it's in the cat cortex. And it follows on a lot of the kind of work that Sanji talked about. It uses, in this case, looking at orientation columns, but specifically orientation tuning across cortical layers in the cat cortex. 9.4 Tesla, 0.25 millimeter isotropic resolution, gradient echobolt, and using a contrast agent ferrium. So these are CVB-weighted images. And you know that you can get, even though this is now very high resolution, 250 microns, you can get multiple slices, and you can get these pinwheel representations of the orientation tuning. at this high resolution with CBV-rated imaging. And with many slices, you can in fact look at across the cortical layers and see these column-like structures of the orientations represented across the 6 layers of the cortex. And here are some tuning curves for these, in this case, 8 different orientations, and some data showing, for example, average distance between pinwheel centers across the cortical layer. But the most interesting data that we were is actually this one. This is orientation selectivity index. And in this grant, that is a brain initiative grant, in fact, it was predicted that there's an expectation that the orientation selectivity is not going to be uniform, and that layer 4 likely to be, in terms of what we can detect with hemodynamic response signals, less tuned than the outer layers. And indeed, the CBV fMRI data shows this. The outer layers are much more selective, and layer 4, something over here, is less in terms of its orientation selectivity index. And the equation for this is given over here. Or you can take this data and bin it into more like integrate over some distances to give you different layers like this. And indeed, there are statistically significant differences between L2 and L3 and L4 and L3 in this data. And but at the present time, not only we are interested in this in the imaging with fMRI, but we are also interested in optical imaging. And this is there's a very large optical imaging effort now going on in CMRR. This is the lab. There are 3, in fact, experimentalists, 3 experimentalists working with their own setting up, or have set up their own groups, working only with optical imaging techniques. And of course, there are connections between these labs. This is the Kara lab, Prakash Kara, pictured over here. And he has set up 3-photon as well as 2-photon imaging at CMRR. And 3-photon, of course, because 2-photon doesn't really penetrate enough to be able to look at layer 4 in the cat cortex, but 3-photon can. And here is a 3-photon image from the Kara lab. This is ex vivo with different labels. This is GCamp labeling the neurons on the outer layers, and this is thalamic afferents labeled by VGLUT2 antibody, which you can do ex vivo, but not necessarily in vivo. And this is something called third harmonic generation, which is something like a diffusion image, optical analog of diffusion imaging in MR, which can actually show you the fiber structures. This is a composite image, as you can guess. But here's the layer structure. It's about 2 millimeters in this case, going into the white matter. And the expectation was that we should be able to reach layer 4 in 3-photon imaging in the cat neocortex. But there's data from recent data. This is again ex vivo to identify where layer 4 is with the VGLUT2 marker. And then here's the trace. So layer 4 in the cat cortex is, something like 850 micron or 900 micron deep. And but here's some data. This is now not neuronal data, but still vessel data, either from arterial or capillaries, looking at in the arterioles, vessel dilation. There's a select, example vessel from layer 2-3 at 234 micron depth, and layer 4, 900 micron depth. And these are the orientation presentations, and you can see that there's some sort of a difference here. And again, this is now blood velocity measurements in the capillaries. Here's an image of the capillaries at Z-plane of about 830 micron from Dura. You cannot get this by 2-photon. It cannot reach deep enough, but 3-photon does. And here's your orientation selectivity index, layer 2-3 and layer 4. And you can see that there is, in fact, as the prediction was set in the grant proposal, layer 4 orientation selectivity index is less compared to outer layers. And this is the comparison between optical imaging, red and green, and gray, the fMRI. So there's really very, very good correspondence. And we are very pleased and excited with these results. And of course, there's still a lot to do. We have to get, these are vessel responses now in the fMRI, in the optical imaging. And then we are interested in pursuing, of course, neuronal and synaptic responses. And then there will be bold in the cat and bold in humans. Now, bold in the cat, to be able to do this, whether spin echo or gradient echo is tough at this kind of resolution. But there are some techniques that I will show you that will enable us to do that as we go forward. And the technique that I want to show you, there are a couple of them, is this one. We haven't really talked about this very much. This is something that we are all very excited about. This is work by Luca and Steve Muller, Logan, Mehmet Akçakaya, Isa, and myself. And Steve and Mehmet has developed algorithms for this image processing that has enabled us. And Luca and Logan has been doing a lot of the experiments and analysis of the data. So it's suppressing the thermal noise in fMRI. As you know, when you go to high resolution, you are dominated by thermal noise. This is the noise that actually ultimately kills you, not the physiological noise when you are doing columns and layers. So we follow the paradigm that actually following on the footsteps of an experiment that was done previously in CMRR by Amiram to measure the point spread function of the bold response. And the same paradigm, you have a target presentation and a surround presentation. You have multiple presentations like that, and you're looking at what is coming out as a result of these in the human cortex. Here are some images. This is 0.8 millimeter isotropic and SMS multiband acquisition. One slice from the many slices that were acquired, coronal slice, and they are somewhat noisy, as you can see. And I'm sure many of you who are doing this kind of work would be familiar with that, because of the fact that SNR at 0.8 millimeter isotropic resolution is not that good. But here's a new image reconstruction. It's called the Nordic reconstruction. Now the noise is suppressed quite a bit compared to this, as you can see. Here's another slice, subject 2. Another slice in another subject. And this looks somewhat better. You can actually see that there's quite a bit of cleaning that goes on with this Nordic reconstruction. So now you can actually repeat that paradigm I showed you. You can obtain data with 1 run, 3 runs, 5 runs, and this is the standard reconstruction that is coming out of the scanner. And of course, it increases the t values. The t statistics gets much better, and when like for example in subject 2, you can hardly detect something. At 5 runs, however, you can see the response for the target and the surround. Very easy to distinguish it. So it's one, target is red and surround is blue, and you can see that. But here's the Nordic reconstruction. It's single run. Single run. Now, single run here is approximately equivalent to something like 3 runs, and single run in this case equivalent to something like 5 runs. So in fact, this Nordic processing is meant to suppress thermal noise, and it increases the t statistics significantly. And you can say, well, we may be getting something of the order of factor of 3 to 5 in terms of the amount of data you need to actually get the same t statistics. And these data, I won't show you that much, but it doesn't change the percent signal change, but it does change your ability in repeated runs, the accuracy with which you can actually acquire that, measure that percent signal change. And the most important is that you can do that without actually blurring the image. You know that you can, of course, always blur the image by applying some sort of a filter, and you will improve the signal to noise, and you will get better T statistics. But of course, in blurring the image, you've lost the information that is necessary to map columns and layers. But this method really does not blur the image at all, as you can see from before and after, or rather without Nordic and with Nordic, essentially, the full width half maximum of the voxel definition remains essentially the same. So then, well, can we go to very high resolution with these data? And yes, here's an effort, again, experiments carried out by Logan and Luca, and 0.5 millimeter isotropic resolution data, 7 Tesla gradient recalled epi, and multiple slices. Well, they look pretty noisy, as one can guess. You look at the image, very noisy. It's hard to say anything, especially if you go away from the very surface of the cortex. And here's Nordic reconstruction. Yes, wonderful. You can actually start seeing that, well, you can do functional imaging with that kind of data. And here's Nordic 1 acquisition and standard approximately 10 acquisitions. And you can say that here maybe more than 10 acquisitions have to be averaged on the standard reconstruction to reach what Nordic does. I should say that, you know, when you go to the effect of this reconstruction will depend on your signal to noise. For example, if you have very low resolution and your thermal noise is much, much more dominant, you will get a better effect than if you were, for example, doing very large voxels, in which case this the reconstruction may not even have an impact on your fMRI data. And of course, you cannot go down forever. At some point, you don't have any more information, even for Nordic to pull out from the image. But 0.5 looks very doable. And time courses, standard reconstruction, you can hardly see anything in there but noise. But here's the Nordic reconstruction, very nicely delineating the responses all the responses of the visual stimulation. And when you construct the images, Luca has worked very hard. These are hot off the press to supply me these images recently. I mean, he collected the data very recently as well. They look very noisy. This is target versus surround, but with Nordic, you can now actually see this area. So I think 0.5 at 7 Tesla is really reachable at the present time, or has been reached, if you like, with image reconstruction. And you can ask the question, are there additional gains possible? And indeed, yes. I mean, this is really very exciting in my mind, that not only there are image reconstruction gains, but there are many other possible gains that are coming down the pike that will actually improve those things. You can go, what are some of these gains? Well, you can, of course, go to high resolution imaging, but high resolution imaging always requires acceleration unless inner volume selection is used. So that means that you are always going to have noise amplification to this G factor in this data. And the data I showed you, 0.5 millimeter, 0.8 millimeter, have this G factor noise amplification. And you can go to higher number of receive channels to suppress this G factor amplification. And we've demonstrated that relatively recently, a paper published last year, when Eddie Auerbach built a 64-channel receiver for our 7 Tesla scanner. And then we were able to get some 7 Tesla 64-channel data to compare with the coil that everybody uses with 32 channels made by Nova. So these are actually G factor noise maps for 2-dimensional acceleration, 10-fold 2-dimensional, 16-fold, 14-fold, 16-fold, 14-fold, and 12-fold accelerations. The G factors are presented in a sagittal view is a maximum projection, maximum intensity projection projection to an axial view. And you can see that this kind of acceleration even is a little too much for this coil. You get, in some spots, threefold amplification of the noise. In many areas, twofold amplification of the noise. So it is actually marginal, if not absolutely non-usable. But here's the 64-channel data. You can, as you suppress this noise significantly at this case, and you can even think about 12-fold two-dimensional or even 14-fold acceleration. So G-factor noise is in our human data all the time. It's real noise, and we can suppress this by going to higher channel counts. And there are other image reconstruction approaches than what I showed you besides Nordic, and they are complementary to Nordic. Here's work by Steen and Akçakaya, mainly Akçakaya lab, accelerated imaging using deep learning neural networks. This is an image reconstruction approach. And here are some human images, twofold, fourfold, sixfold, and eightfold acceleration. You see that normal reconstructions, what most everyone uses, you can do twofold, it's acceptable, fourfold, marginal, sixfold, and eightfold, unacceptable. But here is a deep learning, it's a physics-guided reconstruction from Mehmet's group, showing that, well, fourfold is beautiful, and even eightfold is acceptable in terms of acceleration here. So again, suppressing G-factor noise, if you like, reconstruction noise. But real noise in all of your fMRI data, unless you are using very, very, very restricted field of view, in which case you are not using parallel imaging probably. So of course, we can go to higher fields and higher channel count receivers. For us, this means 10.5 Tesla. We've been working very hard on this, and really the primary reason for doing 10.5 Tesla is, of course, my passion to be able to do robustly column-level imaging in the whole brain in the human, but we do other things with it as well. But here's an effort. We have to build a lot of coils for this. This is currently our 32-channel coil with a 16-channel receiver. And unfortunately, at 10.5 Tesla, we have to go through FDA to get our coils approved. And this is a major endeavor. It's a whole physics research, actually, about how you do this. But so this coil is not yet approved for human use, so I won't be able to show you human data, but I think we are getting very close to it. But I can show you phantom data, human-like phantom with electrical properties that match the human head. You can see that there are significant SNR gains. These are SNR data going from the NOVA coil, where we are, many of you and us are doing 7 Tesla experiments. This is this NOVA and 7 Tesla with 32-channel receivers. This is our 32-channel coil. Well, one channel was not working at that time, so it was 31-channel coil, and SNR data measured at 10.5 Tesla. You should ignore the fact that there are some areas missing here because they did not use both of the rings of the transmitters, so that some areas did not have B1 for excitation. But the SNR calculations are independent of this B1 excitation, and they show you actually going from 7 to 10 and from NOVA to our coil, you get something like factor of 2, and in some places even as much as 3 gain. So that's another major gain that we can build upon the data that I showed you with 0.5 millimeter resolution. Kamil, but we are going to higher number of coils, Kamil, and 128 is being built right now, as well as the 128-channel receiver being implemented by Eddie. And we expect signal noise to increase, of course, in the cortical regions, not so much in the center of the brain. But you have to remember that, again, if you want to cover a large part of the brain, if not the whole brain, you always have to accelerate, and then the center of the brain especially suffers signal noise loss or noise amplification for the G factor. So 128 or 64-channel coils really improve that. So accelerated imaging is not going to look like this. It's going to look like SNR is going to increase everywhere in the brain. And so let's come back to essentially which approach should be used.
Kamil,
This is Seiji also talked about this gradient echo or spin echo. Vaso, a lot of data coming from, beautiful data coming from Vaso. CBF, Seiji showed that CBF is very accurate, and CBF, I showed, also talked about how orientation columns are very accurately mapped by CBF. CBF is, in the human, is low SNR application for columns, so nobody has attempted it. But with some of these things, maybe CBF comes back into the question. And I'd like to maybe even say that among these, there is quite a bit of potentially, even though we've done a lot of work with spin echo, maybe there's some life for gradient echo still in this development. I mean, there are, for example, there are efforts that showed, well, you know, even though you have big vessel contribution in the gradient echo, when you look at layer images, and if you are using models to describe your responses in the brain, as Michel Morel paper has shown, that you can extract actually information from the gradient echo data across the layers. There are other approaches like Kamil Uludağ, who showed that you know, you can actually model again the vascular response across the cortical layers and actually extract information despite the vessel, the large vessel contribution. And there is one paper just came out from our group. This is work by Kendrick K. In fact, I think it's also not also. It's, I think, very promising in terms of decomposing the data that is coming from gradient echo in terms of large vessel and small vessel contribution. So essentially, it relies on the fact that vessels have somewhat different temporal responses. And here's a, we can just look at this picture here. Essentially, there's an early group of responders, and there's a late group of responders. And he came up with a very, really elegant method of separating these responses and eliminating the, or looking at them separately, if you like. And here are some data. For example, here's the standard reconstruction. This is layer data going from different layers from depth 1 to depth 6. And you are seeing, of course, as you know, very bright areas that come from vessels. You can see them in the in these anatomical images here. Here are the vessels, for example, showing up. They correspond to these very bright spots. And of course, in deep layer, as the Polymeni work has shown, you have the vessel contributions are much less. And this is his deconvolution of the early and the late responses. The vessels are ending up here, and the early responses are without the vessel. I want to explain this a little bit, that this is really very nice, because you may have something like this, for example. Let's say that this area, the blue one, is your voxel. Here is your vessel, and the bold effect going beyond the vessel boundaries significantly. And you may have this vessel traversing through the voxel, or you may have the vessel actually outside of the voxel, but the bold effect from this very large vessel may even penetrate into the voxel. But the idea is that then you can actually, in this case, the signal coming from these voxels will be some sort of a combination of the microvascular contribution from bold and the macrovascular contribution from bold. And the very nice, beautiful aspect of Kendrick's work is that you can actually eliminate this, but you retain this in the this information. This is very different, for example, with what you would do if you were to just remove those voxels which are suffering from the blood vessel, large blood vessel effect, in which case the whole voxel is gone and you lost the information about the microvasculature in that voxel. So like in this scenario over here, if you were to just cut off the layers where the macrovascular signal is contributing, that information is gone. You cannot really do layer fMRI. But in Kendrick's, this information is retained. So that is the, you can eliminate this, but you can retain this. So all in all, there are a lot of things that are happening in the human area which are exciting. They can be brought together. You know, at 10.5 Tesla with image reconstruction, deep learning, or Nordic and new coils, etc., we will go way beyond 0.5 millimeter isotope, 0.1 microliter isotropic. And when you go to that kind of a resolution, I want to also point out, life becomes very simple. I want to point out also this using, in fact, optical imaging from Amiram Grinvald. And here you have an optical imaging image with all the vessels, and this is orientation, ocular dominance columns. And you can see these columns, and of course, even though this is a differential image, you can see the vessels as well. And is it a problem? Yes. Is it a major problem? No, because the resolution is high enough so that you can look behind, in between the vessels, and you can see actually what is happening. So high resolution, in fact, solves a lot of your problems, all with respect to the discussions we've been having. With that, I would like to end and acknowledge my many people. I've acknowledged a lot of them during my talk, but this is our engineering group who actually indispensable for CMRR's function. The image reconstruction group. I don't have a picture of a lot of the people, but I have these pictures from a couple of years ago. Of course, many people have, as I mentioned, contributed this work. So with that, I'd like to end, and I apologize for my very long lecture, but I hope that at least you saw some interesting data that may make it worthwhile. Thank you very much.